Hi, folks. If you could please turn off your video and keep your video off as well as your microphone, and we'll start in just a minute. All right, we're approaching 40 participants here, Sylvia, so I think we should go ahead and start. All right, welcome everyone to the Hatfield Marine Science Center virtual research seminar. Thank you for joining us today. My name is Yi-Chung Chung. I'm the Academic Programs Manager at the Hatfield Marine Science Center. And today we have a special guest, Sylvia Yamada from the Department of Integrative Biology at Oregon State University. I wanted to welcome her. Sylvia Yamada is a marine ecologist with research interests in salmon stock identification and life history and range expansion of intertidal invertebrates, including non-native species. She has monitored the abundance and recruitment of the European green crab to Oregon and Washington estuaries for over 20 years and authored the book Global Invader, the European Green Crab. And with that, I'd like to introduce Sylvia.
Well, thank you very much, Yi-Chung. I'm glad to be here. And first of all, I'd like to acknowledge my co-authors, Jennifer Fisher and Mike Cosgrove from Oregon State, and Sean Schooler from South Slough National Estuarine Research Reserve. Each year, I always look forward to working with the students in the marine biology class and in the marine invasion class to sample green crabs in Yaquina Bay. And for many years, we did not get a lot of green crabs. And I was always happy when I just had a few to show to the students. But that all changed in the last few years. After the last El Nino. Many of you may have noticed that green crabs are now abundant enough and large enough to enter sports gear. So you get a lot of calls from people trying to figure out what are these animals in my traps. So hopefully I will be able to, I tell you what's going on. Okay, let me see. There we go. All right. Yeah, green crabs are a global invader. They're native to Europe and North Africa, and they're found on all the continents now, except Antarctica. And what's to note about their distribution pattern is that there are two isotherms that are linked to critical life history features. Number one, green crabs need at least 10 degrees Celsius in order for the larvae to develop. They need that temperature at least for part of the year. If it never gets above 10, you're not going to get green crabs. Likewise, there's an isotherm at 18 degrees Celsius. So green crabs need to experience at least 18 degrees for part of the year for the females to successfully incubate the eggs. Now you can see that all the native and introduced locations are within the temperate zone. Sightings in Brazil, in Madagascar, in Sri Lanka, in Burma, in Hawaii, none of these took. My main emphasis for this talk is talk about the abundance pattern in Oregon estuaries. But first of all, I'll give you a little background on ecological impact, life history, and the Northern Range expansion, and also talk about my future goals for next year. As you all know, green crabs are predators and ecological engineers. And whenever green crabs are abundant, be it in their native habitat or at other locations where they've been introduced, they can have a tremendous effect on the prey distribution. When they're abundant, they can prevent cockles, mussels, and urchins from forming forming beds. And the best example of the impact of green crab has been the decline of the soft-shelled clam industry on the East Coast during the 1950s, when there was a warming period, much like there is now. Also, they can select for predator resistance in some of their prey species. This little littorine, Littorina obtusata, used to have a very thin shell and had a little spire. But after the green crabs were introduced to the East Coast, they developed a more predator-resistant phenotype. They're thicker shelled and they have almost no spire at all. They're almost round. Now, the impact of green crabs as an ecological engineer has been documented recently on the East Coast and also maybe in British Columbia. When green crabs are abundant, they can turn a very productive eelgrass meadow into bare substrate. And they do that through their foraging activities. They take their appendages and they sift through the sediment, tearing up the plants and cutting off the stipes and the roots. Now, eelgrass is a foundational species. So when the eelgrass is gone, a lot of species lose their shelter and their food source. Now, green crabs are a fan-shaped crab, much like a Dungeness or a red rock. But if you look at the spines behind the eyes, you can only count 5, where the others have about 9 or 10. They are medium size. They don't exceed 4 inches or 100 millimeters. They're not always green, or green is not a real good diagnostic feature, because if you look at this little female here, her abdomen is green, but she's also starting to turn a little yellowish in the rest of her underbody. And this male here is red-orange. So it's just a natural process between molts. So the proteins in the carapace are becoming denatured, much like when you boil a Dungeness crab, it turns red. All right. They attain sexual maturity when they're about 35 millimeter in carapace width, and they need about 15 degrees Celsius before the gonads mature and they can mate. This little female here is carrying a nice little clutch of eggs. She will aerate the egg sac and also clean it, and then when the eggs are ready to hatch, they hatch into zoea larvae. And these little zoea 1 larvae, they rise to the surface and ride an outgoing tide into the near shore, where they undergo vertical migrations and feed on plankton. Now they go through a number of zoea stages and finally into the transitional megalopal stage, which means that the animal can swim and also crawl. So this megalopa, when it's ready to settle, it will again rise to the surface and ride an incoming tide back into the estuary, where it will settle high on the shore amongst the marsh vegetation. And then it will metamorphose into a miniature crab. Now, in 1998, there were a lot of green crabs that were in our estuaries, and during winter break, a number of students from fisheries and from zoology joined me, and we worked at the Hatfield Center, and we tagged a whole bunch of crabs, green crabs, trying to get at their growth rate. And we inserted this little tag between the upper and lower carapace in that suture line. That's the suture line that opens first during molting, and then the crab crawls out backwards, so that a tag will be retained through the molting process. And by July, we recovered a number of crabs, and you can see they all molted, and the smaller ones had molted twice. Okay, using tag data and also following the growth of the strong 1998 cohort, we were able to come up with a growth curve for green crabs. And it's remarkable, similar for Oregon, Washington, and British Columbia. So the diamonds here are for averages for Oregon estuaries. These are Washington, and the triangles are individual crabs that were found in British Columbia. So you can see that initially they grow quite rapidly. When they're small, they molt every week or two, then a little less. But by the end of their first growing season, they were about 48 millimeters in carapace on average in all in Oregon and in Washington estuaries. Then in the spring, they start molting once or twice. The following year, they molt maybe once. And then after that, maybe once a year or not at all. And then after 6 years, I have the impression that they all died of old age, because I found some dead crabs in the field. And also I took some crabs, some larger crabs, into the lab and wanted to do a feeding experiment. But these crabs did not feed. They were very sluggish, and then they died for no apparent reason, probably old age. Okay. Now, Johan Hjort, a famous Norwegian fisheries biologist, made the observation that whenever there was a really strong year class of commercial fishes in the North Sea, then it could be traced back to ocean conditions when these fishes were larvae or juvenile fish. And so it is for green crabs. One is able to predict the year class strength from ocean conditions during the winter. So green crabs need three things. They need a larval supply, be it a local larval supply or from far away. And in order for that larval supply, the source population has to experience at least 15 degrees Celsius for their gonads to mature and for them to mate. And secondly, when the larvae are ready to hatch, the water temperature needs to be 10 degrees or above for the larvae to complete their development. And finally, they need favorable currents that take them back to habitats that are suitable for them. Now, a lot of these conditions are met during strong El Nino events. And as you all know, we had the 1997 to 98 El Nino, which was the largest of the last century. That was a big one. And also, most recently, the 2015 to 2016 El Nino was an equally big one. And during these El Nino events, you have warm water along our shore, right here, indicated by the white and the red. Also, the currents were very strong during the 1997 to 98 year. The currents were strong in a northerly direction starting from September of 97 to April of 98. So there was a big window of favorable currents to take the larvae northward. Plus, they were unusually strong in January and February. The currents averaged about 50 kilometers per day, and drifters that were released off Newport made it to Vancouver Island in no time at all. And thirdly, because the current is moving northward, there is a shoreward component due to the Coriolis force pushing the larvae into the estuaries. So El Nino events are really important for the success of green crabs. Okay, so how much can they move northward during an El Nino event and during a non-El Nino event? If we take velocity of about 10 kilometers per day off our coast, and we estimate that the crab larvae spend about 2 months in the plankton, then in a non-El Nino year, they can travel over 300 miles, very likely from California to Southern Oregon. That'd be no problem. And in an El Nino year, the distance can be 5 times as great. So 1,500 kilometers. So they could actually move from California, the larvae could move from California to Vancouver Island. All right, just a quick summary of the Northern Range expansion. Green crabs arrived in San Francisco Bay probably during the 1980s, maybe earlier, and it's believed that they were transported along with bait worms from Maine. They were wrapped in Ascophyllum alga, and young green crabs could have been hitchhikers on that alga. It wasn't until 1990 that scientists actually became aware of green crabs, and by then the population in San Francisco Bay was quite large. We had crabs of all sizes, including gravid females. Then from there, they spread by way of larvae in ocean currents, primarily during the winter time, during the Davidson Current, and they made it to Bodega Bay in 93, 95, Humboldt Bay, possibly also Coos Bay. But it wasn't until 1998 when we saw seeding of all the Oregon, Washington estuaries and Barkley Sound on Vancouver Island. From Barkley Sound, it didn't take very long at all for the crabs to move northward, because leaving Puget Sound, the San Juan de Fuca current is very strong, and it's outward into the ocean, and it hugs the shore of British Columbia. It's called the Vancouver Island Current. So that current flows north year round, even when the upwelling starts further offshore. So in a very short time, they made it to the tip of Vancouver Island, then 5 years later to the mainland, central coast, and then last year they were found on Haida Gwaii or Queen Charlotte Islands. Now, we always were worried that green crabs might get into the inland Salish Sea. And for many years that didn't happen because the current in the surface water is outward for most of the year, except during winter storms when can be a current reversal, and it is on the US side of the strait. But usually the winters are too cold. It's below 10 degrees. However, 2015, 2016 El Nino, there are 2 years where they could have made it into the Salish Sea. And here we are in 2016. I'm with a group of volunteer green crab samplers on Westcott Bay on San Juan Island. And guess what? I happened to be there the last day of August, and they found the very first green crab in the Washington Salish Sea. It was a male that had recently molted. It probably was in the second year of its life. And a couple weeks later, smaller crabs in the first year of life were found in Padilla Bay. Here is Westcott Bay on San Juan Island, and here's Padilla Bay. And since then, you can see that green crabs are found throughout the Salish Sea now, especially the last few years. And what's disturbing is that around Bellingham and about the Canadian border there in Blaine, there seem to be new recruitment, maybe new year classes that might have been locally produced. So we're waiting to see whether the green crabs are establishing in the inland sea. If they do, it's going to be really bad news, because there are lots of habitats where they can thrive. There's lots of clam and oyster culture going on, so there's plenty of food and plenty of sheltered bays that are where red rock crabs and other large predators are absent. So they're just going to take off if they're actually, established by now. They would spread throughout the inland sea. It's not good news. Now, I mentioned how ocean indicators, temperature and currents can aid the range expansion of green crabs. Ocean indicators can also control the local abundance in Oregon, or estuaries, Oregon and Washington estuaries. 1998 was a huge year for green crab recruitment in all our estuaries, including Grace Harbor and Willapa Bay. That year class lasted for about 6 years, and then they died of old age. So given that information that we had an El Nino in 1997-98 winter, had a strong year class and no strong El Ninos after that, the prediction was that green crabs would go extinct in Oregon. That was my prediction. Well, I'm almost right, except that there were little blibs here, 2003, 5, 06, 2010, we had a mini El Nino, and then we had complete recruitment failure here. I should mention that this shows young of the year green crabs. And these are crabs that are after their first year of growth on the mud flats. And we're having catch per 100 traps. So right here, you have 1 per trap right here. This is a 98 year class, a strong El Nino. Then here, 2015, we have the second strong El Nino. And in between, we have a few places, a few years when green crabs did recruit moderately well. But look what's happened lately. Recruitment every year. 1, 2, 3, 4, 5, 6 good years of recruitment. All right. So where are the green crab larvae coming from? Well, there are multiple sources. At first, we found that California must have been the source because that's where they came from. Looking at these diagrams, courtesy of Bill Peterson, it shows the climate, ocean climate in Oregon in the winter and in the summer. So in the winter, we have currents, winds pushing the water northward and also towards shore. And along with that current, there's warm water coming from California with a community of copepods like this pseudocalanus, which is indicative of warmer waters from California. All right, and it appears that green crabs must be traveling in this warm water from California as well, because these copepods, these southern copepods, are good indicators of green crab recruitment strength. So the later the winter conditions persist, the greater the chances of larvae, of green crab larvae, to make it into Oregon and settle in our estuaries. Sometime around April, plus or minus a month or so, we have spring transition. And that means that the winds shift. They start blowing from the north, pushing the water southward and offshore, bringing up the cold water, nutrient-rich water during upwelling. And during the summer, the community of copepods is characterized by northern copepods, such as Calanus marshalli, which has a very big oil sac. So these northern copepods are very nutritious, and that's what salmon thrive on. So the more upwelling condition, the better for salmon, but not for green crabs. So Jennifer Fisher and I, and Mike Cosgrove, we put together a multifactorial analysis of all these ocean factors that we thought would predict green crab recruitment strength. And in fact, they do as individual regressions. Each one of these is significant. And what you notice is that there are 3 indices that we use that are related to temperature. The Oceanic Nino Index, Pacific Decadal Oscillation for March, and the winter temperatures at 20 meters. Then there are those indicators that of northern transport of larvae coming from California, along shore currents, along our coast, physical transition. The later it is, the better it is for green crabs, southern copepod abundance, and northern copepod abundance, which are related negatively. And this is the graph that principal component analysis came up with. The principal component, number one, explained 67.5% of the variability. As you can see, it gets warmer this way. In this segment, this quadrant, it's warm. And in this quadrant, it is also, the currents are coming from California. So, but if you remove the last two values for 2018 and 2019, we get a much better principal component one. 77% of the variability is now explained by ocean condition. So something's been happening in the last few years that our model is starting to break down. Our model that green crabs are coming from California when the water is warm, and and we have lots of southern copepods. Okay, so just to illustrate this a little more clearly, I will focus on just two of these ocean indices. We'll just look at winter temperature by itself, and we look at southern copepod abundance. Okay, so here is that same graph of catch per 100 traps for young of the year green crabs in their first year of life. And we superimpose temperature, winter temperature. And you notice that 10 degrees is sort of the cutoff point. Below 10 degrees, the green crab larvae just don't develop. They're not successful. And you can see whenever we have a big peak, the water is warm. Here, 2003 to 2006, it was warm. But here, 2004, we don't have a big response in green crab recruitment, mainly because we had a very early spring transition. But 2010, a mini El Nino, it was warm. And then 2015 was warm. And look what's happening though. Even though the temperature in the subsequent years has gone down all the way down to 2020 to below 9.5, the green crab recruitment strength has not gone down. So something is going on that's different than it was before the last El Nino. Okay, let's just look at plain temperature. And you can see that ocean surface temperature at 20 meters is a pretty good regression, explaining 61% of the variability, interannual variability. But look what happens when we add the last 3 years. 18, 19, and 20. They're way out of line. They should be down here. But the R squared is now only 34%. Something is happening that's not the same as before. All right, let's look at the biological indicator, southern copepods. Now, so the southern copepods are not good salmon food, but they are good indicators of water masses coming from California, and presumably green crab larvae are traveling in these water masses along with the southern copepods. Very good correlation here, southern copepods and green crab year class strength. But what happens when we add the last 3 years? This is 71% of the variability explained by the regression, only 43%. These 3 years are outliers. Any ideas what might be happening? I think some wheels might be turning. Well, I got my first clue when I was trapping green crabs with Christina Geierman in Coos Bay. Christina is a high school teacher and also a marine biologist, and she told me that she found a lot of small green crab molts along the drift line. I think it was 2016. And okay, so 2015, we look at the size frequency distribution right after the 2015-16 El Nino, and we get a nice distribution. Green crabs range from 30 to about 55. No small crabs. There were no There was not a breeding population of green crabs prior to this date. In 2018, the size distribution of young of the year crabs looks quite differently. You see that there are a lot of smaller crabs that we did not find in 2015. Okay, another way to look at this a little bit more clearly. In 1998, that was an El Nino year, no green crab populations in Oregon estuaries. We get the same kind of distribution, 30 to 55. 2015, another El Nino year, very, very similar size frequency distribution of the young crabs. Again, both years, no breeding population. But then we look at 2016, 2017, 18, 19. We are picking up smaller crabs along with the smaller molts. The crabs would be too small to even enter our traps. So where are those smaller crabs coming from? Any ideas? Well, it could be an alternative larval source. We all know that green crabs larvae come from California, at least the initial ones. That was the main reason why we had those nice relations with ocean conditions. They came from California, the larvae, during the winter. But also, we now have evidence for self-seeding of Oregon populations. And also, I'll show you evidence of receiving larvae from the north. Next slide. Ellen Shanks at Oregon Institute of Marine Biology has been studying Dungeness crab recruitment for years. And he uses light traps. But unfortunately, none of the green crabs ever entered his light traps. But there was one year when he had a grant. And it was very fortunate that the year was 2010. That was a mini El Nino. And he had a grant to look at the plankton of Jordan Cove for a whole year. And what he found was that in January, February, and March, he found first instar green crabs. And where could they have come from? Not from California. They were too young for that. They had to be locally produced. The water temperature was warm enough. It was above 10 degrees. So we can have local production of larvae. That has been shown. Next evidence, Carolyn Teppold from Woods Hole Oceanographic Institute has been studying the genetics of green crabs for probably 20 years now. And she found that there was one population on Vancouver Island in Souk Harbor that is genetically different from all the other populations on the whole West Coast. So that population, believe it or not, was introduced by biologists unintentionally. They were collecting mussels on the West Coast and they were parking them in Souk Harbor for a short time just to keep them there until they were ready to analyze them for toxins. That's how we think a population got started in Souk Harbor. And that population is genetically distinct, it's inbred, and it's the only one that in the north here that is different. And there's the ecologists at Macaw Bay and I here at two sites here, we're supplying Carolyn with samples of young of the year green crabs. And what she found was that one crab from Macaw Bay and one crab from Tillamook originated from Souk. It arrived directly from Souk as a larvae. And one crab in Netarts Bay was a hybrid. Now, isn't that interesting? So now we're looking at catch per trap for all the crabs that we found that in these 4 bays in Oregon. I did not trap last year, but you see that nothing much was happening until the last El Nino and the population grew up. Here, Sean Scholar, he sampled in Coos Bay, and these are his numbers. And you can see the last 1, 2, 3, 4, 5 years, green crabs have steadily increased. And what's disturbing is that in some sites, the crabs average over 10 per trap. And that is sort of considered sort of the guideline for or cutoff point for detecting ecological effects. So above 10 per trap, you might be able to document green crab effect on the biota. So that's very disturbing, especially the trend is increasing. So what have we learned? Well, now we have 6 strong year classes in Oregon. There's evidence for local reproduction. And if that is the case, then we have a local larval source for another 6 years. We also have larvae from northern populations. Now, the Sooke population is just one of many on Vancouver Island. So we could be receiving quite a few larvae from Vancouver Island, especially would happen after spring transition. They have, for right now, I learned that we have La Nina predicted for the rest of the winter and then possibly shifting to neutral conditions in the spring. So that's good news. And also, I learned that the marine heat wave has been dissipating. So cooling should be good news, right? Well, it used to be good news before because, you know, if you had cold water in the winter, like in 2008 and 2012, you had recruitment failure for green crabs. However, look at this one. Look at 2020. Water temperatures during the winter were cold, and we would not have predicted any, right? But look what we got. We got green crabs recruiting that year. So obviously, something else is going on, and most likely it's larval sources other than from California. So my goals for next year are threefold. It's quite obvious that we should be doing more intensive sampling of the incoming cohort, the young of the year green crabs. We should target them earlier, more often, and also different locations in the estuaries. And I'll be working on that with Sean Scholar. Sean and I will be preparing some of these crabs that we're catching for genetic analysis. So Carolyn will have a chance to find out if any of them are coming from Sooke on Vancouver Island. And thirdly, I've been working with Jord Hardage of the University of Hull in the UK. Jörg is a chemical ecologist who has been working on green crab pheromones, mainly the female sex pheromone. And we had some promising results a couple of years ago. This time, he has male sex pheromone, which should be more promising because it would attract females, and it's the females that we want to remove from the population. Also, he tells me he has a scent deterrent that would keep green crabs away from an area. Now, that scent would be very useful for shellfish growers to keep the green crabs out. So that's the end of my talk, and I'd be happy to take questions. First of all, I'd like to acknowledge all my recent co-workers in Oregon, Washington, and British Columbia. Thank you.
Thank you, Sylvia. Great talk. So I would like to open up the floor for any questions. Anyone online that would like to ask a question, you can ask via over the video or also in the chat. I know I see that Brett Dumble earlier mentioned that he recognized that they were a local source, and he said that they over the hump for adults in estuaries. So basically they were able to, sounds like, anchor themselves in the estuary. Let's see, a lot of thank yous from folks. Any questions though? Any questions from folks online? Oh, okay. So Rick Pinnell has asked, any sampling in Nehalem?
I had a port sampler from ODF&W who did sample in Nehalem when he was doing, I think he did it for 2 or 3 years. He's no longer there, so I have not been sampling. I was the only one sampling north of Coos Bay this year, so I was only able to do my usual estuaries. I didn't include Nehalem, but Nehalem shows the same pattern as the other estuaries. So the pattern that you see in one estuary, you find in the others. A good year in one is a good year in all the others.
John Chapman says, great talk, Sylvia. Did the sex ratio of adults change?
I do not think so, no. No.
Okay. Liz Parati from ODF&W is asked a question, any chance we have evolution of larval tolerance to lower temperatures?
Well, that could happen. I think Catherine de Rivera is here. I don't know if she wants to answer that.
I don't know that I'm great at answering this one, but I do know that on the East Coast, a cold tolerant strain had come into Canada, and then that traveled down the coast, and something like that could be happening here too, that sort of there's colder adapted ones, whether from a secondary introduction or just from time up in BC that then came down here, or they could appear. The variation is there for sure. And certainly there's a lot of difference among the genetics for green crabs on this coast based on what our temperature tolerance. That's what Carolyn Tupolt's data show.
Thank you, Catherine.
And thanks for the great talk, Sylvia.
Thank you.
Yeah, a lot of thank yous online. Brett Dunball asked a second question. Great talk, Sylvia. Any change in distribution, for example, in intertidal, subtidal, or habitat occupied now that they are abundant?
I've heard that in British Columbia that they're actually going to the open coast now. I don't think they're abundant enough here to do that, but I get a lot of reports from divers from the Eugene Dive Club. I've talked to them twice now, and they always keep telling me that they're at Siuslaw, there is they call it the crab hole, and they find them in there, and it's pretty deep. It's the mouth of the estuary. So there's no reason why they can't go deeper. Yeah. Okay. They do have more predators there, but they could, they can survive there if they can stay away from the red rocks, red rock crabs.
Kevin Book has a question. Can you relate any recent examples of medium or large scale ecological effects?
That's a tough one. I think they've identified some ecological effects in California earlier. They've been linked to the decline of the shore crabs, Hemigrapsis oregonensis. I'm not sure if we have any concrete evidence that they are impacting any other species right now. For many years, green crabs were just so rare, nobody paid any attention to them. And but now I think we have to switch gears and actually look for some effects. And I think Sean is doing that in South Slough. He's looking at eelgrass bed effects, what the green crabs might be doing. Yeah.
We have an alum, Jose, has a question. Jose Maranjaran. Question is there any lag in the correlation between temperature and recruitment?
Temperature and recruitment. Well, you have to realize that I don't really measure recruitment in the ecological sense. I'm measuring recruitment in the fishery sense. When your species of interest is actually entering your gear. So what I'm looking at is winter effects like temperature and current patterns, and then looking at what is the density of or catch per unit effort of green crabs at the end of their first growing season, because that's when I can get enough of them to sample. So that I don't know if it's a lag effect. I think you need warm winters for the larvae to survive. And if it's below 10 degrees, the larvae just don't make it. So I don't know if it's a lag effect. I think I would not call that a lag effect. It's just the fact that my sampling is lagging behind because I cannot measure those little larvae. I'm not equipped to do that.
He has another question that goes with that, and that is there evidence of distance from the mouth correlation?
I've been finding green crabs even younger the year, all the way up to Johnson Slough, by the oyster farm. And when we were doing, when I was working with undergraduates over 20 years ago, we found them at Creticer's Landing, which is even further up the estuary. But yeah, they're found throughout the estuary. They're not found in Toledo. I think it's too fresh there.
Okay. Sarah Hinkle has another question. And it is the 0 crabs in the traps in Coos Bay in 2015.
Oh, that's because I didn't sample that year. I think I went on a trip.
Okay.
I went to I think I went to Cuba or something, and I didn't get to sample all the places.
Okay, that explains it. She thought it might be a sampling issue, and obviously.
No, it wasn't a sampling issue. It's just that I couldn't be in too many places at once. That's before Sean came on board. I was the only one that's sampling all the Oregon estuaries.
Wow. Okay. A couple more questions here. Christina DeWitt asks, you indicated green crab can reduce softshell clams. Are there impacts on native crab species? And I think you touched upon that a little bit.
Native crab species on green crabs?
Actually, green crabs on native crab species. Impacts on native crab species.
Well, the evidence that we have is that red rock crabs can really do a number on green crabs. They have much more powerful claws, size for size, and also adult Dungeness crabs are not, will attack green crabs. What's happening on a smaller scale when crabs are younger, everybody has to be small sometimes. So I'm sure green crabs can eat small red rocks, and in addition to small shore crabs, small Dungeness. So anything that's smaller is fair game, and they're also cannibalistic.
Okay, and then looks like we've got a couple more comments, but I don't see any questions. Some comments are Brett Dumbald says, you said now over 10 crab per trap per day here. He can't remember, but what was that number in California estuaries back in the 90s, and have those numbers changed in California?
I'm not sure about California. Again, Catherine de Rivera might know more.
Yeah, the numbers have changed in California. They oscillate around right now. They're in general down, especially in places on the southern part of their range, like Elkhorn Slough. The numbers are way down. There's several factors that could be causing that, and we're not quite ready for that publication. But one interesting thing is sea otters might be one of the reasons. And let's see. So elsewhere, they're a little lower. At times when they were at their peak, we definitely got more than an average of 10 per trap. Some traps were full, like no more crabs could get in full.
Wow.
So we don't want that here.
No, we don't.
No, we don't.
Maybe we need some sea otters in here.
That's not all they eat, though.
It's true. All right. Well, it looks like there aren't any further questions. Sylvia, thank you again for gracing us with this talk and your presence. Thank you, everyone, for joining us today. What a lovely day it was. And I should probably share next week's seminar topic, which is survival of the fattest, integrating photogrammetry measures of health into population assessments for killer whales.
Wow.
John Durbin from the Department of Fisheries and Wildlife at OSU, and also senior scientist with the North Gulf Oceanic Society, will be giving a talk next week. All right. Oh, and a couple more. Oh, and everyone says thank you.
Thank you. It's been a pleasure.
Wonderful. Great. Well, Sylvia, this went really well. We will share the recording with you. It will be made available, as with all of our seminars. They're recorded and they will be on our website. And I'm sure either Cinnamon and I will share that with you. I also wanted to throw out that if you needed assistance with collections over the next, well, actually from here on out, let me know. I think I might have some folks who can help assist with helping you trap crabs.
That would be great. That would be great, Ishang. Wow.
Yeah,
Yes.
that's really, it's a lot of work to do.
Well, it's nice to have help. Yeah.
Okay.
Yeah.
Everyone says thanks. Great
Been a
job.
pleasure.
Thank you. Thanks, everyone. Sylvia, if you have any questions, feel free to send Cinnamon and I an email if you have any questions.
I'll take you up on the students. Yeah.
Yeah.
That's cool. Thanks.
All right. Take care.
Bye.
Bye-bye.